Good day, class. I hope that you're doing well today. So as you recall, during our course orientation, that the subject is divided into two. So you have your medical mycology and medical virology. So for the first term and second term of the semester class, we will try to cover your medical mycology. And then for the last term finals, we'll try to cover your medical virology. So medical mycology class is actually 4% of your board exam under your microbiology and parasitology. So anong ibig sabihin nito, class? If you will take your board exam soon, there is 6 major subject class. So sa 6 na subject na dun, class, isa doon si microbiology and parasitology. So sa 100 na tanong doon, class, 4 or 4% ang lalabas sa medical mycology. So sa 4 doon, class, 2 lalabas yung sample collection, transport, and processing. Tapos yung 2 tanong naman, class, lalabas ay culture pagdating sa medical mycology. So our course attack naman, class, in terms of this subject is that we will try to focus more on the clinical sampling collection, transport, and processing, as well as with the culture. Tapos, lagyan din natin ng clinical manifestation pagdating sa fungal infection. Okay, para at least after you take this course, you're ready for the 4% of the microbiology and parasitology board exam. Okay, so the primary goal of this video lecture class is first, at least you will be familiarized with the mycology terms kasi magkaiba yung mycology terms natin na ginagamit pagdating sa bacteria, parasite, and or viruses, magkaiba din sa mycology. Second class is at least we will know the basic fundamentals of mycology, specifically what class? Specifically the general characteristic of your fungi. Kasi if you will recall, nung nag-start tayo ng bacteriology, we start characterizing your bacteria, like what's the classification of bacteria, gram-pass, gram-neg, what's the morphology, etc. So, ganun din pagdating sa mycology class. So, we'll try to characterize and differentiate your fungi from other living cells like plant cells and or animal cells, as well as with your bacteria and viruses. So we will try now to start discussing the basics. So the term mycology, class, is derived from the Greek word myces, meaning mushroom. So literally, class, pag sinabi natin mycology, it's the study of mushroom. Pero pag ganito ba yung definition, class, ang pag-aaralan ba natin is mushroom? No, class. Take note that mycology have different division, class, and one of the division, class, is medical mycology. So ito yung focus natin, class. Pakitake note na lang din, class, in 1910, Raymond Sabrod, class, published his book, Letini, which was a comprehensive study of the dermatophytic fungi. And with this accomplishment, class, he is also regarded as the father of your medical mycology. Familiar with the name, class, Sabrod? Okay? Diba pagdating sa culture media, this is one of our culture media, class. Ayan. Okay, culture media, tawag natin doon SDA, class, Sabrodextrose agar. Okay, which is culture media for the isolation of saprophytic fungi. So for the general properties of your fungi, class, fungi generally are eukaryote. So ano ibig sabihin ng, class? That fungi and animal cells have almost the same cellular components. For example, they have Golgi apparatus, you have your nucleus, and they have your mitochondria. However, class, your fungal cells can be differentiated to your animal cells with the 2 components. So, ano yung dalawa, class? So, yung dalawa, class, is that the fungi contains ergosterols, class. Take note with this one, paki-highlight naman. And then, second, class, that all fungi possesses cell wall made in chitin. So, take note with this one, class. Okay? So, pagdating sa bacteria, the cell wall component is peptidoglycan. Pagdating sa mycology naman, class, the major component is your chitin. Yun. So, take note with the 2, you have your ergosterol and then you have your chitin, class. So, they have a rigid cell wall because of the chitin and therefore non-motile, class. Take note with this one, non-motile. a feature that separates them from the animals. Okay? Compare also with bacteria. Bacteria, they have motile and non-motile. So, pagdating sa fungi naman class, madali lang. Madali lang siya i-memorize. Bakit? Kasi may general rule. Okay? All fungi are non-motile. Next class, now we should differentiate your fungal cells from your plant cells because your plant cells are also part of the eukaryotic family. So, how can we differentiate this one? So, fungi class, they are chemoheterotrophs. So, yung ibig sabihin nito class, they require organic compounds for both carbon and energy source. And pakitake note na lang din class that they don't have your chlorophyll. Compare with your plants class, plants, they have your chlorophyll. Your fungi does not have chlorophyll. So, yun yung difference ng plant cell at saka ng fungal cell. Additionally, class, they obtain nutrients as saprophytes. Ibig sabihin, class, live off of decaying matter. And or parasite, live off of living matter. Pag sinabi mo saprophytes, class, because your fungi, they are saprophytic, ibig sabihin, class, they are ubiquitous in nature. So, if the environment provides a good nutrient for them to grow, so they can easily cultivate. So, for example, may bread, tapos the bread contains a lot of nutrients, and then the environment provides moisture, so therefore, the fungi can cultivate and grow. So, yun yung tawag class saprophytes. Pag sinabi naman natin parasite, we all know that this term already class, that parasites are like bacteria, viruses, and parasite itself, that they can actually infect humans and then cause infection. So, all fungi class require water and oxygen class. So, pakitake note to class, all fungi, they require oxygen. And there are no obligate anaerobes. Madali lang, di ba, pagdating sa fungi? Kasi pagdating sa fungi, they are all non-motile. They are all obligate aerobes. Compare with the bacteria, kasi pag bacteria, ang daming requirements. So, pag sinabing oxygen requirement, may anaerobic, aerobic, facultative anaerobe, merong microaerophiles. Pagdating sa fungi, very simple lang class. Yan, so take note with this one. So, pag fungi, okay, may dalawang characteristic na tayo doon. Una is that they are non-motile because of the rigid characteristic of their cell wall component, which is your chitin. And then second class, since they require oxygen, so therefore they are all obligate aerobes. So let's jump with the classification of your fungi class. So meron tayong dalawang classification of your fungi. And first, we will discuss based on sexual reproduction. So, based on sexual reproduction naman class, they are actually classified into 4. So, first class, you have your Zygomacetes, which produce through the production of your zygospores. So, zygospores class, in microscopic appearance, they look like this one. Second class is Ascomycetes, which produce endogenous spores called their ascospores in cells called asci. Okay? So, asci class is the plural form of your ascus. So, yung ascus class is a sac form. Ito. Okay, sa loob ng sac form naman niya class, they have a different ascospores. Ayan, ito yung laman niya, ascospores. So, the ascus will hold and make intact ascospores. Yan. Third class, you have your Basidiomycetes, which produce exogenous spores called your basidiospores in cell called basidia. So the basidium class or basidia, this one, this is your basidium, okay? Or plural form is your basidia. So pero meron tayong basidium class, this will be your parent cell. And then they will try to produce sexually by production of your basidiospores. Ito siya. Okay, so these are your basidiospores. So the connecting filaments with your basidium and your basidiospore class is your sterigma. Ito So take note with this one class. Okay, sterigma connects your basidiospore and your basidium. Your basidium is the parent. Your basidiospore is the daughter spores or cells. And lastly, class, based on the sexual reproduction, yung apat natin class, you have your Deuteromycetes or fungi imperfecti. So fungi that are not belong to the mentioned above class, so they are classified as fungi imperfecti. This is a heterogeneous group of fungi where no sexual reproduction has yet been demonstrated. So apat yun class, huwag kakalimutan. Una, Zycomycetes, Ascomycetes, Basidiomycetes, at yung pang-apat naman class, you have your fungi imperfecti. Next class is you have your asexual reproduction. So asexual reproduction is not part of the classification of your fungi, pero since na-discuss naman natin class yung sexual reproduction, might as well touch also your asexual reproduction. So basically class, pag sinabing asexual spores, they are produced following mitosis. Whereas sexual spores class, they are produced following meiosis. So we all know these fundamentals already class. So first class, I will discuss the production of sporogenesis. Okay, first class is the sexual spores of your Zycomycetes, which are known as your sporangiospore, form within the sac. So you have your sporangiospore class, this one, form within the sac called your sporangium. So merong sac form, ito class, tawag naman dito class is your sporangium. Sa loob naman ng sporangium class, you have your mitotic cleavage of cytoplasm, sporangiospores. Yan. Pag may mitotic division na class, ang tawag sa kanya sporangiospores, which is nasa loob ng sporangium. Yan. So the sporangia class are born on special hyphae called sporangiospore. So ito yung sporangiospore class. So ito yung humahawak ng sporangium. So your sporangiospore holds your sporangium and your sporangium holds your sporangiospores. Okay, take note with that one. Okay, so the formation, so this endogenous process of spore formation within a sac is known as your sporogenesis. Next class is the production of your conidia. Okay, so conidia arise either by budding of conigenous hyphae or by differentiation of preformed hyphae. So these develop following mitosis of a parent nucleus and are formed in any manner except involving cytoplasmic cleavage. So take note with this one class, except. Okay, except involving cytoplasmic cleavage. So meron tayong dalawa class. Okay, the production of your conidia is classified into two, either by blastic or by thallic. So pag sinabi nating blastic development class, the conidium begins to enlarge. Okay, and septum is formed. Okay, here the conidium originates from part of a parent. So anong ibig sabihin nito class? So if you have a parent cell, this one is your parent cell, then they will try to form a bud. So that's the budding form class. So if you can observe, from the parent cell, they will try to bud. Okay, so that's the budding form. So they form a blastic. Okay, in thallic media naman class, okay, thallic development naman class, thallic mode of development, the conidium is differentiated by septum before its differentiation. So, ano yung ibig sabihin nito class? So, one parent lang siya class na magde-develop and then they will form a septum. Okay? They will form a septum. Okay, so meron tayong tatlong classification class. Either it's italic and solitary. So, ganito yung itsura niya class. And or arctic. Ganito yung itsura niya class. Mas, thinner compared with your solitary. And then, alternate naman class because merong alternate ito. Okay, so septum, alternate, septum, alternate, septum, alternate. Okay? So, yun kaya siya tinawag na thallic mode of development. So, the cell that gives rise to a conidium is called your conidiogenous cell. So, you have your conidiogenous cell class. Okay? Okay, conidiospores are specialized hyphae. Okay, that bear conidia or conidious cells. Okay, so we'll use the term conidia class. So, the conidia class, this one, okay, so nag-hold ng conidia class, you have your phyalide. Okay, your phyalide holds your conidia. And then, your conidiospore holds your uh, phyalide. Then, phyalide holds your conidia. So, take note with this one class ha. So, ang hinahawakan ni conidiospore, si phyalide. So, ang hinahawakan ni phyalide naman class, si conidia. Yan. Next classification class is based on morphology. So, meron tayong dalawang classification under this one class. So, you have your yeast form and then you have your mold form. So let's start discussing your molds. So, the most molds have a fuzzy or woolly appearance because of the formation of your mycelia. So, if you can observe this one class, meron siyang fuzzy or woolly appearance. Okay? So, pag tinanong sa exam, bakit sila may ganitong appearance, it is because of your mycelia class. So, mycelia class are divided into two. So, meron tayo aerial mycelia or vegetative mycelia. So, aerial mycelia extend above. So, if you can observe on this one class, they extend above the surface of the colony and are responsible for the fuzzy appearance. So, if you can observe on the culture media, ganito yung itsura niya, class. Ayan. Okay? So, pag tinanong sa exam, bakit ganito yung itsura niya? It's because of the aerial mycelia. In addition, class, aerial mycelia supports the reproductive structure that produce conidia. And conidia, in many cases, are used to identify different fungal genera. So, if, for example, this one class, this is a typical characteristic of your Aspergillus species. And then different species of your Aspergillus have a different pattern for your conidia. So, for example, we have your Aspergillus niger, Aspergillus fumigatus, and etc. But take note, class, for mycology, conidia alone cannot be solely used to identify species level of your fungi. So, the same with your bacteria class. For example, if you can see gram-positive cocci, hindi naman din natin pwedeng sabihin Staphylococcus aureus. Diba? So, dapat mag-run tayo ng biochemical test, like for example, MSA, coagulase test, and etc. To at least identify your species level. It's still the same principle with your mycology. So, the microscopic appearance class often aids in the identification of your molds. Okay, take note with the term class, aid. So, ibig sabihin class, tumutulong lang si microscopic appearance to identify your genus level of your fungi. In some species class, they produce antler hyphae, bracket hyphae, rhizoids, and or spiral hyphae. Okay? When we say rhizoid class, this is very common also in microscopic appearance, they look like this one. Para siyang root-like class. Ayan. May root-like siya. Ito. Okay? So, pagdating sa exam, if you can observe that there's a root-like, so yung structure naman niya class is rhizoid. Next class, you have your septate hyphae and sparsely septate hyphae. So, what's the difference between the two class? So, the common between the two is that they are septate. Okay? The difference between the two class is the manner of the cross walls or the septum class. For example, pag septate hyphae class, the pattern of the septation is constant. So, for example, at least almost the same yung septum niya class. Okay? Septum distance. Ayan. Pagdating sa sparsely septate hyphae naman class, okay, have a few cross walls at irregular intervals. May cross walls sa class, compare with this one, meron siyang cross walls, pero in a good interval. Yan, 1, 2, 3. But here, class, you can observe, meron siyang septum, but sparsely siya. Yan. Take note also with this one, class, we can differentiate the hyphae based on the hyaline versus phylloid. Okay, other characteristic useful in the identification of your pigmentation. So if you say hyaline naman, class, kaya hyaline, kaya hyphae are non-pigmented. However, your phylloid hyphae naman, class, are dermaticaceous, so therefore they are pigmented. So pag tinanong naman sa exam, class, bakit sila pigmented? This is due to your melanin. Okay, if you can observe, class, this one, they are highly pigmented compared with this one. They are lightly pigmented because they don't have melanin. This one, class, they have melanin, kaya they are darkly pigmented. Next characteristic that we need to study, class, is dimorphism and polymorphism, class. The term dimorphism, class, refers to the ability of the fungi to exist in two form dependent on the growth conditions. Ano ibig sabihin, class? That the fungi is able to transform into two forms based on the different thermal requirements. So what are the two thermal requirement, class? Okay, the two thermal requirement, class, is you have your mold phase, that's your room temperature around 22 to 25 degrees Celsius. And second, class, is the yeast form or tissue state, which can survive at 37 degrees Celsius. So majority of the pathogenic fungi, class, they are dimorphic. Bakit? Kasi they can live on the environment, and then once accidentally ingested or contacted with humans, class, so humans have a higher temperature, for example, 37, then they can still survive. Yan. So, pag outside temperature, mold phase, pag outside temperature 22 to 25, room temperature, mold phase, pag yeast form naman, class, body temperature. So, ang tanong, class, is, are all fungi dimorphic? No, class. There are some fungi, majority of the saprophytic fungi, they only have one phase, that's the mold phase, which is very visible in our naked eye. So, pag may nakikita tayong, for example, nagdidiking na, for example, food, okay, this one is the mold form, okay, because of the fuzzy appearance, because of the mycelium or area mycelium. So, ang tanong, class, does this organism can cause infection? So, the answer of that question, class, is E question. So, itatanong mo kagad, is the fungi dimorphic or not? Pag ang tanong dun, pag ang sagot dun, class, is yes, they are dimorphic, then they can actually cause infection. Pag sinabing, no, they are not dimorphic, so, ibig sabihin nun, class, pag pumasok sila at 37 degrees Celsius, they cannot survive. So therefore, pag di sila nakaka-survive, it means, class, that they cannot cause infection. Clear, class? I hope it's clear. Okay, so thermally dimorphic fungal species associated with human disease include Pastomasis dermatitidis, Coccidioides immitis, Yaverstoplasma capsulatum varcapsulatum, Paracoccidioides brasiliensis, and then you have Reproductive and Penicillium marnifii. Sorry, class, marnifii. Yan. So these are very common thermally dimorphic fungal class that can actually cause infection to humans. But several other fungi class also possess this ability, but have not been described as an agent of human mycosis. So, take note with the term class mycosis. We usually use the term mycosis for fungal infection. Okay? So, if you can read your reference materials, yung ginagamit na nila doon, superficial mycosis, subcutaneous mycosis, systemic mycosis. So, pag sinabing mycosis, it's a fungal infection. And polymorphic fungi class have both yeast and mold forms in the same culture. But majority of the polymorphic fungi naman class, they are not clinically significant. So it's okay not to study them. Okay? So we're done with the mold form class. So next is you have your yeast. So yeast are single vegetative cells that typically form a smooth, creamy, bacterial-like colony with an aerial hyphae. So if you can observe this one class, in a culture media, para siyang bacteria. Okay? Pero if you will try to observe that one in a microscope, para siyang cocci class, pero large. Okay, large cocci. Yan. Kaya pagdating sa mga students, they usually identified your yeast cells with gram-positive cocci. Okay, but cocci are small. Uh, Yeast forms, they are spherical but large. Yun. So, yeast produced by budding or fission class. So, budding involves maturation of the bud to an independent blastoconidium. So, from the parent cell class, they form a bud formation. Ayan. And then, they will try to release their daughter cells. Okay? So, that's the general characteristic of your yeast cells. So some yeast class, such as Streptococcus and the yeast form of Blastomyces dermatidis, produce a polysaccharide capsule. So capsules can be demonstrated by negative staining method using your India ink and your Negrosine. So if you can observe this one class, the lighter form, that's the capsule. So the capsule itself can be stained by Myrmico Musicarmine stain. But the most common naman class, paki-highlight naman dito class, pagdating ng staining with the polysaccharide, you have your India ink or Negrosine. So lastly, class, for the general characteristic and properties of your yeast cells, class, some yeasts are pigmented. So take note this class, some yeasts are pigmented. Example class, you have your Rhodotorula. Rhodotorula class species produce pink colonies. Naman class, pag tinanong sa exam, bakit siya nagpo-produce ng pink colonies? Because of the carotenoid. Okay, you have your carotenoid pigments. While some yeast naman class, okay, like for example, you have your Phionellomyces, Wernickeae class, and you have your Pedra hordeae. There are Dematiceus. In culture media class, they produce E brown to they produce E brown to olivaceous colonies naman class. Yan. So we are done characterizing your fungi class. So review lang natin konti class. So fungi, they are classified into two. Okay, based on sexual reproduction. Tapos meron tayong apat na classification doon. And then second class is based on morphology. So you have your molds and then you have your yeast. Okay? So we'll proceed to the next part of your topic class. For the pathogenesis of your fungal disease class, okay, take note with the term mycosis. I discussed that one already earlier. So most fungi are saprophytic or parasitic to plants and are adapted to their natural environment class. So always remember class, pag nakarinig kayo na term na saprophytic, they are ubiquitous in nature. So anong ibig sabihin ng ubiquitous? Pag sinabi nating ubiquitous class, they are commonly seen in the environment. So they are free-floating. So that's why class, pagdating sa bacteriology, ang number one kalaban natin is the fungi. Bakit class? Kasi pagdating ng bacteria, if you will try to collect a sterile sample, it should be sterile, of course, right? So therefore, pag may fungal element na nakita, or fungal cells, it's a good indicator of e-contamination. However, class, as in medical technologies, we should know and determine if the fungi that we isolated is a true pathogen or a contaminant. And we will try to know the general characteristics of that one pagdating natin sa different fungal organisms naman class. Okay? Infection in humans is a chance event. So take note with this one class, chance event. So if you are immunocompetent, so therefore very less that you will get fungal infection. Occurring only when conditions are favorable. So for example, very low immune system, okay, taking of like immunosuppressive drugs, or for example, diseases like HIV, AIDS. Except for few fungi such as the dimorphic fungi that cause systemic mycosis and dermatophytes, which are primary pathogens, the rest are only opportunistic pathogens. Okay, kaya class, pag may fungal infection ka, it really means that, okay, either hugawan ka, and or second is that your immune system is too low. Okay? Next class is that human body is a hostile environment and offers great resistance to fungal invasion. And we learned this one already during our immunology because we have a different what class? Okay, different layers of your immune defense. You have your innate immune system, okay, that contains also your normal flora. Okay, some fungi such as your Candida and Malassezia 4 have adapted to human environment are exist as your commensals. But however, class 1, for example, na-disturb yung normal flora mo, these organisms like for Candida will try to reproduce the number of your normal flora, bacterial normal flora, and then can cause infection. Kaya class, it's not advisable to, like for example, maligo ng 5 times a day. Why? Because you are actually eliminating your normal bacterial flora. So pag tatanggal yung normal bacterial flora mo, other organisms, for example, Candida, can actually occupy the space and then it can cause infection. So the complex interplay between fungal virulence factors and host defense factors will determine if a fungal infection can cause disease. So infection depends on inoculum size and the general immunity of the host. So pag sinabing inoculum size class, so ibig sabihin nun, pag madaming organism yung na inoculate, or na expose sa'yo, then it can actually cause infection. As well as with the interplay of your immunity. So to review class with this one, so we have the triad of disease. So meron tayong agent, okay, you have your environment, and then you have your host. So for the agent, of course, generally they have virulence factor, the same with your bacteria. So for example, ability to adhere to the host cell, ability to damage host, ability to secrete mycotoxins. These are the virulence factors that produced by your fungi that can actually attack your host cell. However, your host class have different mechanism to counteract this one. So for example, physical barrier or pH of the skin, and or other components of your innate immunity class. Okay? And then, however, if your host defense factors are bogged down, then therefore, the fungi can actually cause infection to human. Also take note, class, that environment plays a very important role in the interplay of the agent and host infection. So, example, prolonged antibiotic therapy, underlying diseases, age, and immunosuppressive drugs. So, breaking one of these class, for example, breaking one of this one, environment, and then your host factor, your agent can actually cause disease to your host. Yeah. So, we are done with your basic mycology topic class. So, I hope that you learned something with this one. So, di naman masyadong malaki yung adjustment. Why? Kasi you are familiar with bacteriology already. So, madali na lang siya. So, next meeting, we will have our assessment quiz. And then, don't worry class, in our Canvas, I uploaded already your quiz guide, and your quiz guide is a PDF form. Okay, a PDF form supplemented with this PowerPoint. So the quiz guide, around 75% of the quiz will be coming from the quiz guide, and then 25% of this topic class will be coming from the lecture videos. Okay, so I hope that you review the video lecture, especially with the picture, kasi sa quiz, meron tayong mga picture that you need to identify. So for example, ano yung itsura ng ascus, ano yung itsura ng septate hyphae, okay, etc. So those are examples na pwedeng lalabas coming from your video lecture class. So if you have some questions, class, please just drop your questions in our group and or raise it to your class representative so we can discuss that one next meeting, class. Okay, thank you for listening, and I hope that you enjoy and learn something in my video lecture. Thank you, class.